Close your eyes. Take a deep breath. Now, picture the first summer that comes into your mind. When was it? What does it smell like? What does it taste like? Open your eyes and imagine a world where it's possible to capture those smells and flavors of your memory and use them in the kitchen. What if we told you that with the aid of microbial cultures, we might just be able to make that future a reality? Introducing Culture, microbial kits that bring your memories to life. Using cutting-edge genomics research, we are finding new ways to bring life to your kitchen. Our custom kits capture the smells and flavors of a particular time and place. And our recipes help you make your kitchen creations even more unique and delicious with a touch of nostalgia. We have work to do to make our vision a reality, but as we like to remind ourselves, the future always begins as science fiction before it becomes science fact.
Hi, I'm Nikhil.
Hi, I'm Sarah, and we are the Microbial Memories team. You just saw a speculative vision that Nikhil and I imagined, one where we could capture moments in time with the aid of microbiomes and bring those flavors into our kitchens. Today, we want to share with you a little bit about our inspiration and the science that we're building off of.
So first, let's talk about the science. Our vision involves creating a sense of time and place through flavor, and not just through fermentations of food, like what has been done already at the Noma Fermentation Lab, among others, but through a novel approach that builds on some emerging genomics research. We started with a question. How is a food's taste affected by its place? For example, if you tasted enough wine, you know that the same grape made into wine in different regions can taste completely different. Why is that? To begin answering this question, we read the work of Nicholas Bukalic, Susan Ebeler, and other researchers who have investigated the role of microorganisms in wine terroir. Terroir is the complex flavor that encapsulates so much information about that wine in the glass. Where it's from, what climate was like, when it was made, the soil conditions, and more. All of this information comes together in the subtle nuances of flavor that could allow you to tell the difference between two bottles made from the same grape. Could it be that the specific microorganisms present in fermentation are a factor in the development of terroir? Bukalic and team conducted some experiments to explore that question in particular. First, in their experiment, they took samples of pre-fermentation grape must, basically the grape juice, before it becomes wine, and they extracted the DNA of microorganisms present and then sequenced their genomes to get a list of all of the species present in that grape must and their relative abundance. They did this across California for multiple types of wine in different regions, and after analysis, they found strong correlations between the microbiomes in these samples and their corresponding geolocation, climate, and time of production. So in other words, if you were only given this list of species present and their abundances, you might be able to pinpoint exactly where that wine was made, from which grape, and at what time of year. Kind of like how a trained wine taster can do exactly the same just from the taste.
So we had two questions in response to this research. First, could we identify microbial terroir in other types of foods? And two, could we understand how microorganisms contributed to specific flavors? To answer these questions, we designed an experiment to explore whether we could identify a terroir in homemade yogurts that we made in our own apartments. But before we could conduct our research, COVID hit. So instead of investigating the connection between the microorganisms in our own yogurts and their unique flavors, we reviewed literature to learn more about the microbial basis of flavor. We studied research about metabolic pathways, the chain of processes in which microorganisms interact with their environments to produce molecules that we ultimately perceive as flavor. For example, we see tasting notes for wine that say Meyer lemon, wet pebble, and unsweetened tangerine drop candy. We know that these flavors were not added to the wine. Instead, the molecules produced in these metabolic pathways might have evoked those scents and memories for the taster. Given the opportunity to carry out our own research, we would cross-reference the microorganisms in our own yogurt with known metabolic pathways to specify why my yogurt might have tasted totally different from the Nikhil's. But we're pocketing that for future projects when the lab is open again.
If we had been able to do those experiments, we would have shown more evidence for a microbial basis of terroir. And we would have shown how specific configurations of microorganisms in our yogurts could lead to subtle different flavors, depending on where they were made. So we started working from this assumption, and we began imagining what this might have looked like in the kitchen, assuming our research had worked.
Yeah, and that's where our idea for microbial flavoring or a microbial spice rack came in. We imagined taking the microbial terroir genome sequences that we compiled from samples from different times and places and creating flavors from them. We could imagine having a bottle of liquid with a dropper, kind of like bitters, that could be used to kickstart a fermentation or even be dropped on a finished product for an extra touch. Really, we would expect cooks to use their creativity to find new uses for these flavorings, like any other ingredient. Nikhil, you used to be a chef. What time and place would you make into a flavor if you could?
It would be really interesting to add, this whole new set of options into the kitchen. But right now, the main thing that's on my mind is the racial injustices that we're continuing to see in our country. And as I think about the violence inflicted upon the Black American community here, I think about its roots in European colonialization. And it might not feel this way sometimes, but we're only a generation removed from the colonial era around the world. My grandmother, who's alive, was a child during the British occupation of India, not that long ago. And India wasn't even the last country to gain independence. So when I think about what time and place I would want to capture and use in the kitchen, I think about a time before colonial rule. I think about finding ways to capture the flavor of that period. I think about sampling DNA inside of an old fermentation vessel used in West Africa prior to colonization and creating a sort of like flavor of freedom. I don't know what it would taste like, right? We don't know what bacteria would be in that sample, but knowing that those non-colonized species that we found in the sample would produce the flavors in our bottle, that would be enough for me. That surprise would be interesting. Every time we eat food, we're eating our history. And I think it would be so empowering to create an ingredient that allowed us to imagine recipes for an alternate world without the legacy of colonial rule.
I think that's super interesting. And I really would want to taste that and see what that alternate reality would taste like. And I think it's interesting that you mentioned colonization for another reason too. As you know, my research has focused on cultivating microorganisms for space exploration and settlement. And the language we use to describe space exploration often uses the word colonization, like space colonization or space colonies, etc. And those words are so loaded, especially given the sovereign nations and highly capitalized private companies that are the two main forces backing missions. And these are the same two forces that backed colonization in the last millennium. But those words are also used to describe microbiomes, right? Microbial colonies can be diverse, with many species of bacteria, fungi, protozoa living in the same environment. There could be power struggles just like ours, but there can be harmony as well. I don't want to be reductive and overly romantic here, but my broader thought is that we can travel space. As we travel space, we can take inspiration from microbial colonies to study how we can create a manifesto based on diversity and symbiosis from the outset without repeating the same mistakes we've made on Earth. Maybe we could create a diverse library of microbial cultures made with samples taken from people, foods, places all over the world. In other words, creating a microbial archive of human cultures and history on Earth that we can take with us as we explore the cosmos. Other artists have created similar works related to microbes and memories. Cecil Toles, for example, collects smells from interesting places in a city and is able to recreate a city's essence through smellscapes. Also, Juan Wu's project of collecting hand biomes to use in fermentation could be another inspiration. Maybe we could collect these microbial samples and create a microbial broth, to borrow a term from Alison Cutland, that could remind the astronauts of home.
Yeah, I think all of these would be really interesting, right? Creating new flavors, creating a way of archiving memories, and creating a new future as we settle space. And I guess as we reach into the past and project into the future, one thing seems pretty clear. We need a diverse set of voices to help us imagine a better society, build on curiosity, empathy, and respect for life in all forms. Macro and micro, right?
Yep.
Neither of us are scientists by training, but we're committed to pushing the frontiers of biodesign, and we want to hear from many more voices that are often left out of these conversations. So before we sign off, we want to highlight a couple of organizations that are doing work to expand opportunities in STEM. We're going to put these up on our slide, so please go to these links and consider supporting these organizations. Thank you so much for listening, and we're excited to answer some questions.
Thank you, everyone.